Hello, good afternoon. We welcome you for this first session of support events that try to accompany Module 2, to which you have inscribed. I'd like to welcome my colleague Rob Stanley here.
Hello, everyone.
Who's going to join the session with me, and we are trying to give you a little bit help with doing the assignments, but also hopefully make it a little bit easier for you to work with the materials as you go along the sessions of Module 2. So for those who are not used to the web sessions using the GoToTraining software, I'll try to explain how the control panel works. So I first switch on a functionality that helps you see our screen here. And there's a couple of things of notice that you need to be aware of when you're trying to use it. So this control panel on the top left or top right, depending how you open it, will have an orange arrow. If you click on that arrow, the window will be diminished, and it means you have more space on the screen for materials to look at. Right, so below that, a symbol with a an orange hand on a blue background is a functionality that helps you give us a signal that you want to ask something. So there's a possibility, if you have a microphone in your computer or connected to your computer, you can actually join the session via audio, and you can raise our attention if you press that button. And if we pick that up, then we'll try to connect to you and let you ask your question or explain your problem or listen to your comments on the session. And finally, next thing is materials. At the center, you can see a list of files that are attached to this session and that you can, from this materials tab, download. So you can open them and see them, but you can also download them to your laptop or computer. Whereas there are two PDFs, one containing the session slides, and there's another one that contains the paper on which the assignment is based. In addition, as you can see here at the center of our slide are 3 MATLAB files with worked out solutions for the example that's in the materials in the session 1 of module 2, so that you should have worked before the session. The second, called phosphorylation, is the example that we are going to program for you here live. And finally, there's a worked out solution for the assignment. And if you are surprised why you get the solution before you hand in the assignment, well, working with web materials, of course, requires new ways of working, and one of them is you should be at any point be able to look at the solution if you are working alone and if you need some further help and nobody else is there. Because we can't always be online. Sometimes we are, but we are not always online. And in that case, there is somebody to ask. In that case, it's a file with the proper solution. There's also some comments in the text telling what each line of the MATLAB code does, so you can try to infer its meaning more easily. Okay, so much to deal with these things. And we now have a slide of our session, and I start by giving a very short overview. We'll start with a little bit of a MATLAB practice just to get used to using MATLAB fluently, and also to give you some tips on things to do that might not be so explicit from the materials, things that we find useful and we pick up from the forums that people have asked how to deal with. We then do explicit modeling exercise where we derive a model for a given set of data that I'm going to show you in a second. And our central exercise will be then to implement that model that we have derived in MATLAB code let it run and analyze it and use it to obtain the results. So it's an example that you haven't seen in the materials, but it's done in such a way that is analogous to what you're supposed to do in the assignments. So hopefully you can then draw your conclusions on how to use that to complete the assignments, should you have any problems with it. For those who want to know more about eigenvectors and eigenvalues of matrices, because it's a rather abstract concept, there's a video tutorial that tries to make it more intuitive, and you get the web page. So if you download the slide file and you click on that link, you should be able to access the YouTube video and watch it. It's about 10 minutes long, and it tries to explain what eigenvalues and eigenvectors of a matrix in this specific context of the Leslie matrix are in the bacterial aging session. Right. Let's start our session by doing a little bit of MATLAB as a warm-up. So the thing that I'm going to do is I'm going to open my MATLAB, and you can see here the command window. I have arranged it a little bit such that it's easy to read for you. And we are not really going to do things in the command window. So typing in commands, we can do it by saying, what directory are we in? PWD. So I'm in a directory called desktop. But rather, we are going to do something that's much more useful in the long run, especially if you're used to program with MATLAB. We're going to open a new window. And I enlarge it so it's easier for you to see. I'll also switch off the webcam. I've seen enough of our faces. And we now have a space to write a script. So, what is a script? The first thing I can do before I even start to type with it, I save it as something. I save it as a file, in this case in the current directory, and I call it script session 1. So I'll give it some name, make it a script, and I save it. And you might be able to see it on the top right of my screen. It's somewhere there, and it should contain whatever I type in and save will be in that file for later referral. And it means once I have typed a sequence of commands, I'll be able to execute the whole sequence at the same time without having to redo individual lines of the command window. So for the moment, I use it even to hide the command window, and we'll only look at it when there's something interesting to see. So what we are going to do, and we hope this is quite familiar by now that you have entered session 2, is we define some data. We want to make a plot, and I'll show you a few features about plotting using some data. So we produce some test data, and we're doing this using the rand function. Rand is random, and if we want to have normally distributed random numbers, it's called rand_n. So we make a matrix of normalized random functions, numbers, and let's say we have 100 values and we have 2 variables. So we have 100 rows and 2 columns filled with random variables once we execute this thing here. I can also suppress the output by putting a semicolon, and means A now, which is assigned to this outcome of the rand number function, will be a matrix. In order to get a plot from A, I use something called subplot. And if you're not familiar with it, subplot makes multiple windows into one plotting environment. So I want to have 2 subplots, which means 2 rows, 1 column, and I refer to the first one of these 2 by putting a 1 at the end. And then I can just start by, say, plot my matrix A, which means it will plot the 100 random values of the 2 variables at the same time into the same plot. And if I have another way of plotting them, I can look at the two variables against each other, something akin of a scatter plot. Then I take, rather than the first window of the same segmentation, I take now the second window, subplot 2, and I want to plot something that contains the first column against the second column of my matrix A. So I want matrix A in both x and y axis. What is the first column? It will be all the rows of the first column. And what is the second column? All the rows of the second column. So this helps me to plot the first variable against the second variable if we interpret these two entries as variables. Right, so that's enough for an introduction. Let's see it. Doesn't look very beautiful in the second one, so I close it again and make a minor modification, namely rather than just plotting it by the default, I give it an option. I use symbols, so the dots are not connected, and if I make them red, it will be even more prominent. And this is what the plot then looks like. So you can now see our plot. And if I go into the interactive graphic window, we know that this is now can be subjected to changes. So one frequent change is, for example, to change the font size of the axis labeling. It's now if we want to have it much bigger, say 30 points, and we just have to do these changes here. And we can make the lines with which the data are plotted even more prominent by making them larger. So instead of 3, we can make them 6 points. Now they will be thicker, perhaps also easier for you to see. So I've made a couple of changes in the interactive window. Now, in order to avoid having to do that over and over again, it's a very simple trick how you can keep all those options that you have done here for further reference. So this is what I'm going to show you. Under the file bar in the top left corner of this interface, you will find the possibility to generate code, which means all the code used internally to produce this figure, if you click on it, will be available here. So you get it, it opens a new window, and all the code that's necessary is here. So for example, the font size of the labels is specified as an option, as an optional argument in this code here. So rather than to have to do it every time again, you can get it from this thing, which you only need to save. So you can keep this code, and rather than call it create figure, say call it seismic, what can we call it? Seismic M2 plot, just to give it a name. I saved it under this name. And we should now call it also this here, seismic M2 plot. And then you have it as a function, and you can refer to
this. It should
this.
be a small m, sorry.
Which one?
It should be a small m for M2.
Small m for M2.
To match the file name.
To match the file name. Let's see, where's my file name? Seismic M2 was a lowercase m. So here we have it now saved as a function file. And the way to refer to this function is, of course, just here. You can now copy this and use this as a command, as a call of the function in your code. So rather than now doing all these things again, we can use a reference to this call, but we have to see what we need to plot. So what did we plot? Well, we remember the matrix used for the first subplot was called A. So I substitute A. And then, of course, the other things is just the same thing that you used to plot. We will now need to go in here, first and second column for the second plot. That's how the function expects you to hand over these things. And we have now a reference to all this code that you have prepared by interacting with this graphical user interface. These things here we don't need, so you can comment them out by using comment in that on the top of this window. So you can see we have substantially decreased the amount of code needed. And what I can do is I can close this code, and if I execute the new script, which is now saved in my file, then I get the code with the new options, namely with the thick lines and the very large lettering on the X and Y axis as you wish. Okay, that concludes our introduction. This is the way of saving time for later. You can then tailor your own graphical options, the options you want to have in your plots, and you don't need to do it over and over again, but you do it via a function call rather than scripting it. Let me close this window down. No, I don't need to save it. And I just delete it so we don't have, I delete both things so we don't need to worry about them anymore. Okay, let's go and do now, as in the second part of our session, do an explicit modeling exercise. Shall we do it again, Rob? Shall we do it together, Rob?
I think so.
Okay, let's take a look. Here's a bit of a motivation. We have some data, and I don't go into the details. You can look into the paper if you want to see more. Just roughly, this is single-cell measurement of the state of phosphorylation of a specific receptor, in this case the insulin receptor, so hepatocytes, for example. The state of phosphorylation is measured over time upon incubation of a cell culture with, in this case, insulin. So it's the hormone that activates this receptor. And what we can see here is a single-cell measurement of the unphosphorylated state of the receptor. And the idea is to measure how quickly or to what extent is this receptor phosphorylated when it gets activated. So they have, at time 0, they have an initial amount of unphosphorylated receptor, which is stable over time. So that would be a straight line at 1.12, whatever the emission ratio is. Once insulin is incubated, we can see here in this data plot, the amount of unphosphorylated receptor is decreased, and by implication, the amount of phosphorylated receptor increases. So while we don't go into the detail of this experiment, this is the kind of data that we want to model, the process of phosphorylation and dephosphorylation of a receptor upon activation. So let's see how we can set up a model of this process. It's going to be very simple, of course. It's just a qualitative argument, but you'll get the idea. We want to do it using a discrete time modeling approach, just as the one that you saw in the population model in the materials of the first session. So let's see. Rob, how would you describe this model? What can we see here?
So the squarish boxes look like it's protein and then phosphorylated protein. And then we've got two rates going between them. So we've got the K1 rate, which is the rate of phosphorylation of the non-phosphorylated form of the protein. The K2 is the rate of dephosphorylation of the phosphorylated form of the protein. So we've got this sort of cycle between the unphosphorylated and phosphorylated protein.
Exactly. We have a simple model with only two variables, two things that we take into account, obviously given by or motivated by the experiment, and we have two processes that relate these variables to each other.
So the variables are the unphosphorylated protein and the phosphorylated protein.
Right. And these processes that connect them make them a simple reaction network, if you want to speak, and this is what we want to describe. And so what we do is we're going to set up our discrete modeling scheme, and we want to enter these two reactions to account for changes in the concentrations of each of the receptor types. Okay, so what is happening here?
So it looks like we're starting with the next step being equal to the previous step or the current step. So xn plus 1 equals xn, so nothing's changing there, correct?
Right. So the idea is that we relate whatever happens in the future, so whatever changes over time, we relate that to the current state. So if xn is the current state, then our next step, so sort of our step into the future, is somehow related on what is actually there at the moment. So, and as you say, nothing will happen if we have this rule because it means it will stay forever the same. So it means we are missing the interactions between the two.
So now we need to, let's start by adding in the K1 in terms of the rate of phosphorylation. So X, which is the unphosphorylated form of the protein, and Y is the phosphorylated. So if we've got a rate K1 times by the unphosphorylated, because K1 tells us the rate at which it's converted from unphosphorylated to phosphorylated. So K1 times by that concentration. So we'll take that away from the phosphorylated and add that onto the, sorry, take that away from the unphosphorylated and add that onto the phosphorylated form.
Right. There's a process that diminishes the population or the number of unphosphorylated protein molecules or receptor protein molecules. And as one disappears, of course, a molecule, corresponding molecule of phosphorylated protein must appear, either concentration or population number, it's both the same. So we have it taken away in the first line in the X equation, and we have it added in the Y equation in the equal amount. So this is our first reaction step. So this means if we have only the first reaction, then this would be the model to describe it.
Yeah.
K being a rate constant, so to speak, regulating the speed with which this happens per time unit.
So this is adding on the second reaction, so dephosphorylation of the phosphorylated form of the protein. So that's basically the reverse before, but with K2 instead of K1. So adding on to the dephosphorylated and subtracting from the phosphorylated term. Oh, and times by the total amount of phosphorylated protein this time, not the.
Right. In this case, it depends on the variable y, because the more phosphorylated protein we have, the faster it will be with the same reaction rate. So we need to make it dependent on this concentration also, and we have K2 times the y at the time n, taken away from the phosphorylated protein, the second line, and added to the first line in the equal amounts. So it means we have now completed all the things that are in the scheme in the lower left corner. All the things that are indicated there by boxes and arrows have been implemented, and that completes, so to speak, our simple model of a process that relates the two types of proteins to each other. And we think that these are the things that need to be there in order to have a reversible way of the receptor to respond to the presence or absence of insulin in that experiment that motivated us. Next thing is now, how can we derive the thing corresponding to the Leslie matrix in a population environment? Now, what do we have here? We have highlighted in red the things that need to go into the matrix. So what you can see, previously we had only xn plus 1 equal xn, but what that means is that actually we multiplied with 1, so there's an entry of 1 going into the matrix, and there's a second entry depending on the amount of xn, namely minus k1. So we have contributions to that part of the Leslie matrix, 1 and minus k1, which depend on the amount of xn. And the entry that relates to the concentration of our phosphorylated molecule, Y, is now plus K2, so we also know what to fill in the matrix there, and equally for the second line. So you can see those are in red the entries to the Leslie matrix, or in this case we can call it a reaction matrix. It's a matrix that summarizes all the interactions going on between these two substances. And we have highlighted it here in this box. So that is the easiest way that tells us how to fill in our matrix with those rate constants when we get into MATLAB. So why don't we just do this? Because this is finished now. We have specified all our variables, all our interactions. We have set up our model in terms of a discrete time update rule. We have said how the system is supposed to evolve into the future according to these rules, and we have specified what matrix is going to now make our transition run. So Rob, do you want to take over here and start with a little bit of MATLAB coding to implement it?
Okay, I'm going to minimize this. So what we've done already is we've got a MATLAB script file, so a file which is on our desktop, with the information that we were just talking about. So here's the original model with the xn plus 1 as a function of the xn and yn, and the yn plus 1 as a function of the yn and the xn. And then the model matrix that we derived from there, so with 1 minus k1, k2, k1, 1 minus k2. So now let's start our MATLAB code. Of course, MATLAB doesn't know what K1 and K2 are, so we're actually going to have to tell MATLAB what we want them to be. So as a start, I'm going to define these. So K1 as 0.1 and K2 as 0.5. And we might even like to put some comments in our code today so that we know what these are, so kinetic parameters. Now we're free to define the reaction matrix. So this is identical to this matrix here, so I have in comments. So just so we remember what it is, reaction matrix in as a comment. And then now R equals, and it's going to be 1 minus K1, K2, and on the next line, as the next row, K1 and 1 minus K2. And so that defines our reaction matrix. Also, so that's effectively set up everything that we know so far from our model. But that's not very useful, and it's quite nice to be able to simulate that. So we're going to do that as follows. First of all, we want to tell MATLAB how many iterations that we're going to have. So we're going to have this command, this variable called iter, to store that we want 30 iterations. Okay. And now we also need to define some initial concentrations. So, so first of all, we want to have enough. A matrix is big enough to hold all of the output of those iterations. And because we don't know anything about it, we're going to initialize it to 0. So concentration is equal to 0, and then that's going to have 2 along the first axis and iter along the second axis. So we've got 2 values 30 times. Okay. The next thing we know is we have a total amount of enzyme in the system. So where the enzyme is the protein. So we've got a total amount of protein in the system. So the total, in this case, we're going to take as 100. So instead of 100%, or there's 100 molecules. And that's going to be the total amount. So there's going to be every time step. There's going to be xn plus yn, and so xn plus yn is always going to equal 100 in our system. And now we want to set the xn to have the totals to start with. So we're going to have initial conditions where xn is 100 and yn is 0. So we can do that as follows. And for the first column is going to be equal to total 0. Okay, so we've set everything up. I'm just going to remove some lines here, and then say just call this setup in a comment, if I can spell correctly. Okay, add a gloss. Okay, so I'm sorry. I'm just going to make sure we can see what I'm writing. Okay, so we've set up our system. We've got the reaction matrix. now what we need to do is simulate it. So I'm going to do an iteration loop. Please tell me if I'm going too quickly, by the way. So in this loop, we're going to use a for loop, and we're going to call the variable I. And we're going to say from 2, because we already know what the first setup is. So that's for I equals. Okay, I'm going too quickly. So just to recap, we have some kinetic parameters that I've defined as equal to 0.1 and 0.5. Okay, we can change these, and we will change these in a bit. We have a reaction matrix, which was defined from our initial model. So from the mathematics we did in the PowerPoint presentation just before, that gave us this reaction matrix. Then I've said we want 30 iterations. I've set up our matrix which will contain our, the results of our iterations. So that's got 2 rows, where the first row corresponds to the x parameter and the variable, sorry, and the second row corresponds to the y parameter variable. I've said that we want a total of 100 molecules of our protein. And then I've set our system so that the first time point, all of the molecules in the system are unphosphorylated, so they're all in the first row, and then there is nothing in the second row. So now we've got the system set up, we're going to start our iteration loop, and so we're going to use a for loop for this. So we need to give it a variable to store which iteration we're on, and we're going to call that I. And since we know what the first iteration is effectively in our system from this initial condition at time 1, we're going to start this at 2. Then we're going to run it to the total number of iterations using this syntax. So I equals 2 colon iter. Okay? And of course we end the iteration loop. In the middle, we now want to set concentration. Both rows, remember the top row corresponds to the concentration of X, so the number of molecules of X, and the bottom row corresponds to the number of molecules of Y. That's the unphosphorylated and the phosphorylated form of the protein, respectively. So we want both, but we want to set those for time I. So we know what our reaction matrix was, and that was R, so set here. But now we need to times that by the concentration at the previous time point. So, and this is exactly as is in the materials, correct, Gerald?
This is as in the materials for the change of bacteria populations.
So the previous time point was given by concentration. Again, we want both rows, but now we want time point I minus 1. Okay? And so this gives us the concentration of time I, knowing the concentration of time I minus 1, and our reaction matrix R. There's one more thing we'd like to calculate, and that's the proportion of the total amount of protein that's in either state. So I'm going to quickly run through another loop to do that. So we're going to call this calculate proportions. So I'm making sure to comment everything because it just makes everything clear when we come back to the code later. Again, we need the first thing we need to do now is to define another matrix which will hold the results of our calculations. So I'm going to call this prop for proportion. And again, it's going to be equal to, it's going to have 2 rows and iter, so 30 numbers of columns corresponding to time points. And I want to initialize them at 0, so zeros to iter. And that's exactly as I set up the concentration matrix previously. Now we want to run another iteration loop. So now for J. Okay, so we can do this the other way around now. Instead of calculating across the time points, it's now also possible to calculate across the concentrations for x and y for the all time points. So instead of doing for I equals 1 to iter, it's also possible for me to do from j equals 1 to 2. So now I'm corresponding the first row and the second row. So j will be 1 and then 2. And I'm going to make those equal to my rows. So now we want to do the proportion of j. So now j comma semicolon. And so this is saying I want to find everything every column from the first row, and then every column from the second row. And I'm going to calculate this from the concentration that we set up before. And again, I want everything from the first row and then everything from the second row. So exactly as in proportion, I want the concentration commands to match how I select the rows and columns. And then because I want to divide everything by the total concentration, what I'm going to do is dot a slash. And now, if you recall from previous discussions, the dot means apply to every element separately. Otherwise, MATLAB tries to do fancy things with matrix division, which we don't want. We want it to divide every element in concentration, the jth row concentration by total. Okay. I'm going to save this, and then we can run this. So hopefully we shouldn't have any errors. And so we saw everything came out nicely. I'm going to hand back to Gerald to do some plotting.
Okay, let's look at the results. And where am I? So first thing is I'm going to assume that you have now been following us up to this stage. And of course, both the simulation of the model and the calculation of the proportions is in analogy with what was done for the bacterial population in the materials. So we're now going to do a plot. And the thing that we want to do is to plot the concentrations. And as I have introduced it already before, we're going to use subplot to do two figures in one graphical plot environment. And we want to have, just as we set up initially of this session, the first of two subplots, and we define it by this line subplot 2, 1, 1. So we just want to plot the concentrations, but because this is the way it is organized, it turns out that we need to use the matrix in its inverted form. And we're going to then add another subplot. So in addition to plotting the concentration, we also want to see the proportions in the second window. So subplot 2, 1, 2. And here we want to see the proportions rather than the concentrations. And similarly, we use this convention to plot it in the right way. And so what we now have, following the setup, we have done simulations of the temporal evolution of both concentrations. We have calculated the proportions for each of the enzyme species, and we have all values stored in either the const vector or the prop vector. So we can execute this, and we can see the result in a plot, which I'm going to increase so that everybody can see what we are doing here on their screen. And this is our outcome, and let's make a little bit of an interpretation here. What we can see that we have started with unphosphorylated protein being as 100 and no phosphorylated protein at present. And then we have had two reaction rates for the two reactions that interact between the two protein species. So when we run the simulation, what we can see is that both concentrations change initially, but then reach a steady state and maintain there. So the steady state we can interpret is an equilibrium state, namely a state where neither concentration changes, although reactions are going on at equal rates. So a steady state where the unphosphorylated protein remains high. So the higher proportion is that of the unphosphorylated protein, which is the blue line in our case. So we can say that data is unphosphorylated. There's a little bit of an error here. It's 1p too many. I'll just correct this unphosphorylated. And I correct this. Green is our phosphorylated protein. And that holds for both plots. So we can see that in terms of concentrations, we are at the order of slightly above 80 in terms of concentrations for the unphosphorylated, and the fraction then above 80%, as we can see in the lower graph. And for the phosphorylated, correspondingly approximately something below 20 in concentration and below 20% in the lower graph. So what this means, this models now our state before any activator, before any hormone is present. Namely, most of the protein will be in the unphosphorylated state and it will be a stable situation. So we are finished with this. We have had the simulation of the model of the first part. We're coming to the second part in a second. And we can now ask, how can we constructively use the theory that we have learned about eigenvectors and eigenvalues? So this will then be our second part. And we call it eigenvalues and eigenvectors. So what do we need to do? And from the materials you might recall it, and if no, we'll show you there's a convention of getting the values in two different variables. So the eigenvector first and the eigenvalue second, if you use a call called eig. This is what we have learned. And so if we take the reaction matrix, subject it to the function eig, which is calculate eigenvalues and eigenvectors, then in this convention shown here, we can output them and get the corresponding numerical values organized in matrix form. Now, what is the largest eigenvector? What is the definition of the largest eigenvector? Well, we have to take a look at what it is. So what I can do, I can execute this piece of code here. And then we can see the output of this function on the left side in the command window. And it shows us that we have two eigenvectors. They're organized in columns. So we have two, but we don't know which one is the largest eigenvector. In order to find out, we have to check the eigenvalues. So the eigenvalues are given on the diagonal of the second matrix, and we can see that one of them is equal to 1, and one of them is equal to 0.4. So it's now easy to find out which one is the largest. Obviously, 1 is larger than 0.4. So the first eigenvector, that one given by the first column of the vector matrix, eigenvector matrix, is the one that we want. So if we want to save it, so we can call it, what can we call it? Vec largest. Where do we get it? We get it from vec, which is the eigenvector, all rows of the first column. So if we execute this, you can see now in our command window, we have the above lines as before, and now the vec largest is indeed containing the two values, the two entries of the largest eigenvector, namely of the eigenvector corresponding to the largest eigenvalue, which is equal to 1. Are we finished? No. If you studied the materials, you might recall that in order to get the steady state values to which our model will evolve, explicitly we need to look at the normalized eigenvector. Now, what is a normalized eigenvector? Can you tell us? Rob is a mathematician.
So we know the two elements of the eigenvector. So we need them to sum to 1.
Aha.
So
if we sum them now, we have 0.98 plus 0.19. So that's much more than 1. So a little bit more than 1, I should say. So what can we do in order to normalize this? We define some new variable called, let's say, largest norm. We only have one, so we normalize it. Now, how do we do that? We take the largest eigenvector, which we have from the previous assignment, and we divide, again, element-wise. Recall that any division that goes by each element needs this period in front of the division symbol. So we divide by what? We divide by the sum of the two. Means, how do we get the sum? We call a function which is called sum, and we run it over the largest. It's actually not spelled with a capital. I have spelled it like this. So if we take the sum of the two elements and divide every element of the vector by the sum, we'll get the normalized values in return. So I include a command which is CLC, which helps me clear the screen in front of every execution of the script. And then I run the script. There's a button called Run on the top. If I run the script, we have the plot again, which you remember from before. I enlarge the plot such that you can see those values nicely. And if we check on the left-hand side with the output of the normalized eigenvector, and the two values that we find is 0.83 for the first variable, that's our unphosphorylated protein, and 0.1667 for the phosphorylated protein. And by comparison, you can see these are the values that the simulation reaches as a steady state. So from the eigenvector corresponding to the largest eigenvalues, we can predict the proportion explicitly. So in the second graph, you have the proportions plotted. 0.83 or 83%, so means the majority of protein will be unphosphorylated. 83% will remain unphosphorylated, means we're describing to some extent the situation where no hormone is present in our phosphorylated unphosphorylated reaction scheme. Right, so that's basically finished what we wanted to do as a simulation, but let's go back quickly and do a simulation of a case where we now think that you have added some insulin. What change would happen if you add some insulin? So what we know is that in that case, the receptor probably undergoes a change of conformation because the corresponding hormone is binding on the extracellular side. And what that leads to is a larger affinity to be phosphorylated. So the ATPases or whatever is waiting to phosphorylate it, say tyrosine residues, is now more affine to actually receiving a phosphate group. And it means that compared to the previous case, the value of K1, or if we had it of K1, will be increased. So if I say I make it times 8, it will be an 8-fold increase of this rate of phosphorylation, assuming that this is what actually happens. So this is our assumption of a mechanism, that it will be faster way of phosphorylating this because the receptor has undergone a conformation change. We can just try these arbitrary values in the moment. But you see the way the script is set up, just executing the script again will do this whole calculation, but now taking different values for the kinetic constants. So if we do that, we get a new plot. And what this new plot shows us is that indeed this change of kinetic constants has led to a different distribution of proteins. Namely, the value of unphosphorylated protein, our blue curves, now goes down lower, whereas the value of the green curve goes up higher. So we have a high amount of final phosphorylated protein. What we won't do here now, but what I will just quickly show you is that, of course, the graph that we have doesn't really correspond to what we saw in the figure. And by knowing a little bit about this thing, we know that means that the ratio and the absolute value of this constant is not yet correct. I've put in arbitrary values. So if we put different values like 0.4 and 0.1, just as an example, I get different curve shape, but this means still the phosphorylated protein is now there in abundance, but we have a way of, say, an exponential decay in the unphosphorylated protein, the blue curve, as we saw it in the experimental data. So it means this is now a thing that still needs to be done, namely a fitting of these parameters to the model, to the experimental data. We won't do this here, but you can see the idea that you can use those values to fit to actual experimental values, and we'll leave it for you to experiment further around with this in the future. Right, so you could see eigenvalues and eigenvectors are useful in that they predict the final state. If you want to know more features, you can actually extract more information from these eigenvectors and eigenvalues. Please watch the tutorial that was linked to in the slide previously, and try to follow similar steps as those shown here for the assignments. I think one of the last things that you were asked to do is to do a bar chart, and we can just complete our exercise by doing it. Make a bar chart of what? Well, we just, of course, want to see the normalized largest eigenvector. So if we run this again, no, it's vec norm, so I've made an error. So you can see by debugging means I get a MATLAB warning, a MATLAB error message that this name that I put in is wrong, and I can correct it by, of course, taking out this comma which went in here by mistake. So if I now run the script again, it should help me do a bar chart.
We're getting an infinite result in our vec norm. I'm not sure why.
Because I haven't run it again. Running it again. Calculate elements of R. No, I don't get the bar chart, so again I have an error. Can we find that out?
So if you look in our MATLAB command window, we're getting minus infinity and infinity. I see the problem. So we're using the wrong eigen value and the wrong eigenvector. So remember before we wanted to get the largest eigenvalue implied the correct vector to use, and previously that had been in the first column. Well, now we're finding it's in the second column. So
okay, and I haven't accounted for this. I have put in the first column. So if we see that the eigenvalue 2, the second value on the diagonal, is the largest one, I can't just use the first column here, but I have to use the second column in that code here. So if I execute it now, we do get the result that we wanted, namely the amount of phosphorylated protein is up to 80% now, means the steady state value is such that the receptor molecule is in its activated form and is ready to start the signaling cascade that's initiated by insulin in the external environment. Okay, so with this final exercise of a little bit of debugging, we can now save our script and close it, and it's now saved for further reference. Later on, you don't need to retype this again. You can just make changes to this command. I will take away my MATLAB window, and we come back on our last slide. Going to the last slide, We finish this by alerting you to the fact that camera again, alerting you to the fact that our next session is on 26th of February, so that's 3 weeks from today. Thursday afternoon, 4 p.m. If you want to join us again, there's a registration URL. You will also receive an invitation. And Rob and I will try to do a follow-up on today's session. There's more on eigenvalues and eigenvectors, not on the Leslie matrix for a model, but actually for data analysis, an introduction to a thing called principal component analysis, a way to meaningfully cluster omics data sets, which is an important thing in many modern biology-related research. I would like to thank you for joining us today. Please let us know your evaluation by filling out a few questions that you see when you leave the session, and there's also space for additional remarks, particularly if you thought it was too slow or too quick. Of course, we can't adjust to everybody properly, but we are very happy to slow down if we get a request to do so, because we don't want to rush it over people's heads. That's not what these support sessions are for. If you like, join us on 26th of February, and have an evening for today, and see you next time.
Bye.